Hello and namaste to everyone. Today I'm going to talk about, or rather discuss about, some of the important as well as critical issue which is going throughout the world, and the world is suffering in day-to-day population, that is about the cancer. Now, we have all heard about the word cancer, and we are very much familiar too in respect to different terminologies like cancer, tumor, abnormal cells, etc. So, related to this particular terms, that is cancer or tumor, we need to understand that how important they are in terms of degrading them, or in terms of remediating them, in terms of curing them at the disease which it is prevailing throughout the human beings. So we'll understand that what is its metabolism, how it is affecting the normal cells, and what are its different routes through which it is growing and growing and degrading the normal cells. Myself, Dr. Suhas Vyas from Department of Life Sciences, Bhakt Kavi Narsi Mehta, University, Junagadh. So we'll discuss about some important points which are related to the cancer. Now we all know that there are various types of diseases which are occurring in today's world. So related to that, if you see, there are various types of diseases. These diseases are are some of them are very important, which are to be remediated or which are to be treated on the primary basis, and some of the diseases which cannot be treated on the primary or diagnosed on the primary symptoms, they are taking a dangerous effects in the human beings. So if you consider the important points which are related to this session or this discussion, that is, what are the statistics which are having throughout the world about related to the disease, and what is the immune system which works through opposing those diseases, as well as what are the oncogenes, and oncogenes are directly related to the cancer cells, and what is its metabolism of the cancer cells. I'm showing you one of the dimension of these metabolism of the cancer cell. So if you see on the overall aspect of the cancer cells prevailing in the world, that is every sixth death, just see the peak at which the cancer has been growing throughout the population. Every sixth death in the world is due to the cancer, making it the second leading cause of the death. So, isn't it very much drastically increasing throughout the different types of diseases? That is sixth death in terms of cancer, right? And it is the second leading cause of the death, other than the cardiovascular diseases. So it has been proved, or it has been statistically proved about by the Institute of Health Metrics and Evaluation, which showed that the lower as well as the upper estimate of the cancer disease may be from 8.75 million to 9.1 million. The lowest is amounting to be the population which is suffering from this particular cancer disease, that is 8.75, to the maximum range, that is 9.1. There may be different types of causes of the death throughout the communities, throughout different locations, geographical conditions. That may be due to the population size, that may be due to the life expectancy, because we know that there are variations in the life expectancy, that is the age at which the people are dying. There was a time that people were able to live about 90 years or 95 years or more than that, but the expectancy of the life has been decreased, and this decrease of life expectancy is just due to different sort of normal as well as abnormal diseases. Out of these abnormal diseases, one of them which is showing its peak, that is the cancer. Particularly in higher income countries, aging population means that total number of cancer death continue to increase. Whenever there is a limit of about 60 years, 70 years, the data shows that the number of death increases due to cancer. So this effect of cancer has increased its death number due to in higher ages. Cancers are defined by the National Cancer Institute as the abnormal cell which can divide and spread to nearby tissues. So the normal cells will not able to live normally, and this will be affected by the abnormal cells, which we term it as a cancer. As this definition suggests, cancer can rise in many parts of the body, leading to a range of different types of cancers. Because we have came across to number of organs which are being captured by the abnormal cells, or which are being inhibited by by the growth of the normally. Hence, these cancer are in different types of organs or in different tissues which are behaving abnormally, and in some cases it spread to other parts of the body through the blood as well as lymphatic systems. Now, looking towards the overall population in the world, if you see the color variation in this particular slide, it shows that continents, different continents, are not able to hide themselves, or not able to restrict or protect themselves to this particular disease. It is showing the data of about 2016, and if you see, the Asian continent now is getting towards the brighter side, that is towards the darker side of prevailing the cancer. In terms of its percentage and countries, the continents, as I showed in the earlier slide, in the terms of its countries, it is all the countries, major countries, are showed in this slide. If you see, by 2016, each and every country in different continents have been drastically grabbed by the cancer disease. India, especially, is being shown in 0.2%, but it is not to be happy at looking at the values of the percentage, because the population is also very much higher. So, if you consider the population, the 2% percentage is also comes to be on a higher side when we talk on the numbers of the people in the whole population. Sharing about the percentages of different types of cancer growing in throughout the world, the population which is suffering from these cancer types, the toppest one is the breast cancer in the women's. So it is considered to be taking the charge of about 0.12 percentage in throughout different types of cancers in the world, which is being grown by the abnormal cells. So the biliary tract cancer as well as the breast cancer, they range to be the top as well as the minimum, respectively. In terms of India, if you see comparison with the world and the Indian population, percentage-wise, and the types of the cancer which are shown in this slide, here also in India, in women, the breast cancer is considered to be on the highest range in percentage-wise, right? So, and the number of people, now here comes the important and as well as interesting fact, the number of people in the world or in different countries, the data shows that there are about nearly 2 to 4 million people suffering from the cancer in India itself. You can see the darker color of on the India, that is compared to that of other countries, it is showing a higher number of population which is suffering from these diseases, that is from these cancer type of different types of cancers. Worldwide, obviously, the number of population is being on the first different type, that is the breast cancer, and in India it is about 5,25,500, more than those numbers, because these data are showing about 2016. So the toppest one here also, it is the breast cancer. Now if you total or sum these numbers of the population which is suffering from the cancer, different types of cancer, you will be very much shocked that these values are going above 10 lakh, about 10 lakh. The population wise, the number of people wise, this disease different types of cancers is being increasing and increasing, starting from the juvenile cancer towards in the adult mature people who are suffering from different types of cancers. The age wise, the age factor, if you consider in the cancer disease, it also has an something important and interesting fact, that is the mid age. For example, if you say 50 to 69 years of old population are suffering more, that means they are being diagnosed on that particular age. No doubt the abnormal cells have been generated at the lower age, that is maybe about 15 to 20 years, maybe 15 to 30 years, but these at this particular age, it is not being diagnosed. On the later stages, on the later age old, that is about 50 years or so, the diagnosis can be showing the abnormality in the cells, and hence the stages of the cancer are just related to the third stage, or on the higher stage. And this is difficult for the treatment process to have the treatment of 100% treatment of the cancer. In India, comparatively to the Indian population, here also the age factor is on the 50th, more than 50 age of the population. That is, the if the person is reaching towards the 50 years, they they are diagnosed by different types of cancer in their body. So almost about 5 lakh number of people have been diagnosed on this particular range of age, that is 50 to 60 years. Now, as I told you that there are various different types of cancer. These cancers are capturing those normal cells, those healthy cells, and they are to be treated. But unfortunately, it is not diagnosed at the proper time. If it is being diagnosed on the primary stages, on very basic stages, maybe say what we are referring at first stage or in a preliminary stage, they are able to be removed, or they can be treated on different therapies, by different therapies. So the slide shows that if you know there is a basal cancer, if you know there is a skin cancer, it can be diagnosed differently by providing different drugs, by providing different therapies, now to create a powerful immune system for treating those cancerous cells. Some of the statistic data which I have presented on this particular slide, that is, there are about 17 million new cases of cancer in 2018. You just see the fact that these numbers are not including, are excluding the non-malignant skin cancer. The skin cancer we are not including in this 17 million new cases throughout the world only in 2018. So in one year, in one particular year, 17 million cases are being diagnosed as the cancer stage, excluding the skin cancer. Out of these 17 million, 8.8 million, which includes about almost 52 percentages in the males, and as well as 48%, that is reaching up to 8.2 million in the females. So the ratio, if you see from male to female, that is almost very much closer to each to these two genders, that is 10 is to 9.3. So if you consider gender wise, the number of population which is suffering from cancer, that is from 8.8 million, or 52 percentage in the males, and 48%. So it is going just near to the parallel number of figures. So this is not taking any restrictions on the gender wise. The world age standardization, that incidence rate item, shows that there are about 204.7 new cancer cases for every year in men. So out of 1 lakh men, there are 204.7 new cases which are diagnosed as cancer, whereas in the females there are 175 out of 1 lakh females are diagnosed as a cancer. The most, as I told you earlier, the most common types of cancer are lung, female breast cancer, bowel cancer, prostate cancer, and accounting all other different types of cancers. These four types of cancer, they itself shows about 43% of new cases in the cancer disease. And if it continues, if this scenario continues, what I showed you in 2018, that is 52%, 48% in females, 52 in males, if this continues, there is a prediction there is a prediction that in 2040 there will be about 27.5 million new cases. The figure is very huge. I'm talking about only the cancer disease. We are not talking about any of the other disease which is not having an abnormal cell. So these are only the abnormal cells which can be predicted, that about people who will be suffering from the cancer will be amounting to be 27.5 million by 2040. If the recent trend continues, this will be a global issue in the near future. This can increase of about 61.7%. What I showed you in 2018, that is 40, 52 and 48% male and female, it has an increase of about 61.7 percentage for the total population, and this has a huge number in 2014 if it is growing in this particular direction. There were 9.6 million deaths from the cancer worldwide in 2019 18 throughout the whole different types of cancer. The other types of cancer which are found in the worldwide, that is liver, lung, stomach, bowel, are most common one, and which are accounting to be on the death side. Now, these were some of the facts where the different agencies showed that this is the figure, that where country or which type of cancer are being predicted, and what is the number of the cancer disease patients. But now we'll go into some of the biochemical part of the normal as well as the abnormal cells. We all have heard that how our body resists towards the antigen entering into the body. The two important types of cells which has the function, which has the duty, which has the responsibility of invading, of those cells which are entering, which we call it as an antigen, and to fight against those antigens, and they have to uh, protect the normal cells. These two different cells which has the responsibility are B cells and the T cells. Now it is their duty to fight against the invaders. These invaders are maybe risky and easily being treated by these T cells and the B cells. Now we are all familiar with the immune system. Just to look over our overview of these immune system, how it works, that is, if you have an immune system, powerful immune system, the T cells and B cells works very much actively. These T cells, which are also called as helper T cells, the cytotoxic T cells, these T cells, the helper T cells especially, they diagnose the cells, maybe by bacteria, maybe viruses which are entering into the body. They diagnose this through the proteins. Now, we all know that proteins are nothing but the long chains of amino acids. Now, these proteins, wherever they are built on the covering of these T cells, they dissociate into the peptides. These peptides are nothing but the markers. They identify that whether the cell is invaded, or it has entered, and it is harmful or it is dangerous to the cell, to the normal cells. They will be diagnosing, they will be scanning those cells, and accordingly they will release some of the chemicals to degrade those invaders, degrade those antigens. So they will produce the antibodies and try to dissociate, try to degrade those cells which are entering into the body. Now, in a normal case, the T cells the T cells has the projection that there is an there is a, normally what happens is, there is a peptide which has the marker, and which is a marker, and the T cells will be sprinkling or will be projecting some of the chemicals onto those invaded cells, which can be harmful, and dissociates into different compartments. These are the invaded. This is the normal course in which the T cells works. Now, in case of the normal cells, when they are muted, after different divisions, there are chances that some of the due to some of the things which are being taken, for example, like smoking, tobacco chewing, etc. All these things can invade can have some of the injuries towards the peptide chains, or say about the proteins, which can be muted. Now this change in the protein can result into the muted cells, or what we call it as a mutation. Now this mutation creates a dangerous effects. This dangerous effects lead to the muted cell, what we call it as a cancer cells. They develop into the cancer, but they behave abnormally. Now they have the tendency to grow, they have the tendency to produce their own proteins, they have the tendency to produce their own peptides. So their chances are having brighter for growing as well as for developing by themselves independently. Then if we have a powerful immune system, if we have a system which can destroy the cancerous cells, which can destroy any of the bacteria or the virus which are harmful to the normal cells or to our body, then why come the immune system cannot degrade these mass of abnormal cells? The reason is that there can be divisions which are being very much quicker compared to that of normal cells. The normal cells always divide at its own time, but on the contrary, the cancer cells has the tendency to divide in very quick time, because they have the tendency to multiply very much faster, because they do not require any of the guidance or any of the orders of the normal cell. They have to follow the orders which are being given by those abnormal cells. Now, in terms of the cancer cells, when they are there are T cells which are available. When they diagnose the cancer cells, because they have generated from the normal cells, because of new mutation, they will not be that much identified that these are not the normal ones. Hence, the T cells, if the immune system is weaker, if it is not showing a powerful immune system, these cancer cells will not be diagnosed, and hence they have no effect of the T cells or maybe the B cells. Hence, the factors which are to be implemented is the powerful immune system. There is always, we all know that there is always a antibodies which are being subjected to the abnormal cells, which fight against those abnormal cells, and the healthy cells are growing. The abnormal cells are not allowed to grow, and they are being destroyed. Now, if the immune system can recognize and eliminate cancer, then why do people develop cancer? There is a question over here. So, if we have T cells, if we have B cells, then why we are able to develop those cancerous cells? The reason behind it is there are immune systems at one time which can become weaker, or which are being suppressed by those abnormal cells. Or it can be also on the other side, the masking of the cancer cells. These cancer cells protect themselves through the peptides. They release those types of peptides which are not able to diagnose, which are not able to scan by the normal T cells. Hence, they skip from getting destroyed by the T cells, and they are able to grow and multiply and divide themselves in a huge mass. So they releases those types of peptides which are not diagnosed by the T cells, and they are escaping to be diagnosed, and they can be easily being growing themselves in themselves. Hence, they inhibit, these T cells are inhibited by the cancer cells, and they are not able to perform their function, perform their duties, which for which they are being formed. Hence, these cancer cells are growing in quicker time, in quicker time. One, and on the one side, they are suppressing the T cells or the immune system. On the other side, they are growing those abnormal cells very much faster. Now, the question may be there that how these cancer cells are developed. The first reason is that there is a sort of mutation which is happening, that is changing the gene patterns, that is changing the nucleotides arrangement. So there may be number of different cases where the mutation happens or mutant gene is being formed. But why it is happening? Now we can understand from this particular slide, that is, the tissues which are being formed by the cells, they have the inhibitors as well as they have the stimulators which can stimulate any of the process. Now, in a normal case, there should be a balance. There should be a balance, that the cells should not generate more amount of concentration of any of the component, maybe any of the chemicals, or the cell should not generate on the lower side of those concentration. Hence, the balance should be maintained. These cells, there is the responsibility or function of these cells not to generate too much as well as not to generate too less. Hence, they have to maintain the inhibitors as well as they have to maintain the stimulators. So what happens is, if there are more number of inhibitors, they will be always be suppressing the other concentrations. On the other side, if you see, there is less generation of those inhibitors. That means they are not able to divide, they are not able to protect those immune system which can target those cancerous cells. So it is to be balanced very much in control, that is pruning to the baseline as well as restoring to the baseline. The normal tendency should be maintained for each and every amino acid as well as protein, carbohydrate, lipid, etc. All these factors should be produced in a normal concentration, that is in the normal balance. Then and then the cell can maintain their life cycle. In both the cases, if the cell is producing more number of concentration of different types of components, then also it prevails to the disease. On the other side, if it is not able to maintain the concentration, that is in the lower concentration are produced, then also it goes towards different types of diseases. Now, if there is an excessive amount of angiogenesis out of different components, it also goes towards the disease like cancer, blinding disease, psoriasis, arthritis, etc. If there is insufficient or less concentration of different types of chemicals produced by the cell or which is to be produced by the cell, then also there are coronary heart diseases, strokes, neuropathy, etc. All these are some of the imbalance created by those chemicals. So the cell have to behave normally, and this normal conditions are depending on the concentration of those chemicals or those compounds which are to be produced by the cells. Generally, when the cell is behaving abnormally, or say about only one single cell has been muted, and it has not divided much, they are not that much harmful. But if it grows, if it divides, if it multiplies in more amount very much quickly, it has a massive effect. Now, this massive effect is obviously on the dangerous side. Now, we may have heard about the term benign metastasis. All these are some of the effects which are considered to be the cell which can travel, the abnormal cell which can travel from one place towards the other place. It has an contagious or infectious property which can also disturb or which can also dysfunction any of the other part of the body. If you consider this particular chain, that is from abnormal condition of these cancer cells which they are starting, then about 5 to 10% of the responsibility is of the genes. Now, out of the different types of cancer, only about 5% chances are there that genes are responsible for those cancerous types. 90 to 95 percentage are just on the basis of the environment in which the population or the human beings are surviving. Maybe diet, maybe tobacco, maybe infection, maybe obesity, alcohol, or any of the other types. So the major part is about the diet. The major part is about the environment in which we are surviving. So if you control the environment, if you have some of the improvisation in the environment, this cancer disease can be decreased. Now, but now we are moving ahead towards how these normal cells turn towards the cancer disease, that is the cancer cells or tumor cells. Now there is a term which is referred as oncogenes. Now they are genes. They are not the normal genes which are supposed to behave or function normally, but they are considered to be the oncogenes. If you consider the normal pattern of our chromosomes, this chromosome is has the starting phase or has the primary component, that is the base pairs, the nucleotides, right? A, T, C, G. We all have came across these terms somewhere in our lower classes. So if you see, these base pairs are arranged in a proper sequence. They are being arranged in a very perfect sequence which makes a gene. These genes form a helical structure as far as human beings are concerned. These helical structure are the double helical structure, which what we call it as a DNA. These DNA are binded with the proteins, which we call it as a histones. These histones bind the DNA in a long chain. These histones binded by the DNA chain is called as chromatin. Chromatin forms the chromosome. So starting from the base pair up to the chromosome, if any of the base pair has been changing its function or has been changing its location, they can be muted or they can be termed as a mutant. So it is the importance of these base pairs as well as genes which are arranged in the histones. They are muted, then the whole protein structure will be changed. So what happens in normal cell? They come together, they form they form towards the active enhancer, and these active enhancer has the tendency to get muted from the neighborhood boundaries and form the cancer cells. Now there, as I told you, that there are different types of nucleotides which are arranged, and these nucleotides, that is the base pairs which forms the genes, but these genes, if are muted, they form the cancerous genes. Cancerous genes. Now there are some genes which are associated with these cancerous genes, which prevails the cancer disease or cancerous cells, are highlighted in this particular slide. I have marked some of them, that is MYC, MYC gene, what is called as MYC gene. They are functioning which is involved in protein interactions. The genes which I have listed over here is some of the genes which are referred as oncogenes. Whenever there is an interaction with the proteins, or what we call it as a various different types of cellular factors, they are the important one. because they perform the function during the transcription, the process of transcription, right? So these transcription are being producing mRNA. Now these mRNA, if they are being muted, hence they will form different proteins which will dysfunction the normal cell. MYC gene. Then there is also one of the gene which is called as P53. Now these P53 genes are very important because they are the genes which suppress the tumor. But if they are not able to function, then they will decrease the process of apoptosis. Now we all know about the apoptosis. Apoptosis is nothing but a normal cell death. Normally the cell has to die at one point of time, so the death of the cell is being inhibited, is being stopped. Hence this p53, if it is inactivated, that means there is no death of the cell, and these cells, normal cells, can be converted into or muted into the abnormal one. Hence this is also one of the important, that is the tumor suppressor gene. Another one are RAS gene, that is RAS. They are the, especially they are the GTPase, that is GTP binding protein, and these are important in signal transduction. These signal transduction complexes help in GTP binding protein. If they are mutated, they will perform abnormally. Hence they are being reported as the abnormal genes if they are mutated. Cis genes, which is one of the growth factors. So all these genes which can be translated into an abnormal condition or abnormal gene, they are referred as the oncogenes. Now normally the term which is called as proto-oncogenes referred to as the normal genes. These proto-oncogenes has to perform a specific function of producing the proteins. We all know that the genes binds with the histones, as I told you in the earlier slides. These histones goes for the production of the chromatin or formation of the chromatin. The chromatin forms the chromosome. So hence these proto-oncogenes, which are supposed to produce the protein or to form the protein, they have to induce the cell division and proliferate proliferate. So the normally the process is in a normal course, that is production of proto-oncogenes. These will produce proteins, the normal proteins. These normal proteins has the function that they have to divide normally, they have to, that means to proliferate, and they also have to inhibit the cell death, right? So the function, the normal function of these proteins are to produce the growth factors. For example, C cis. They also have to perform the growth hormone receptors. Example, epidermal growth receptors, tyrosine kinase, what we call it as a TKR, that is tyrosine kinase receptors. They have to function the growth hormone as a growth hormone receptors. The third one, the third major function is for the GTPase. For example, enzyme of guanosine triphosphate, and the example which I had shown you in the earlier slide in the cancer gene, that is RAS, that is which is related to the protein synthesis, that is the normal protein synthesis, and also the transcription, various transcription factors, which these transcription factors help in the process of transcripting, that is the production of the mRNA. Now, if these transcription factors are not behaving normally, that is, if they are muted, they will also transcribe the abnormal mRNA. One of the best example in the transcription factor is the MYC gene. Now, the importance of this MYC gene is to regulate the gene, that means to control the gene to produce the mRNA, as well as it also codes for the transcription process. These transcription factors are important because of producing the normal mRNA. So I have listed over here the proto-oncogenes. They are the normal genes which produces normal proteins and has to function these many, that is growth factors, growth hormone receptors, GTPase, as well as transcription factors. Now, if you consider these as this as a normal proto-oncogenes that is functioning normally, and if they are muted, you just imagine if they are muted, if the MYC gene is muted, if the RAS is muted, if the epidermal growth receptors are muted, they are not diagnosing the cells, abnormal cells, then the mutation occurs. Now, there are chances that any of these genes which are muted can behave abnormally, can produce an abnormal protein which is not required. So after this mutation of proto-oncogenes, now no longer these proto-oncogenes are called as proto-oncogenes, they are referred as oncogenes. Now the peculiarity of these oncogenes is they also produce oncoproteins. As the proto-oncogenes produces proteins, here the proteins produced from the oncogenes are referred as oncoproteins because they has the capabilities to produce the proteins, but they are not the normal one. They also perform the functions of the cell division, they also perform the functions of proliferation, but they have some of the mutant proteins or mutant genes which do not behave normally. Say, for example, there is inhibition of apoptosis. There is no death of the cell, which is supposed to be at a particular time. Here I've shown one of the example, that is if we have a mutation in one of the factors, that is maybe in the growth factor, growth hormone receptor, GTPase, or transcription factor, because transcription factors, I have considered here the MYC gene, because transcription factor is one of the important point where the MYC gene produces mRNA. If it is muted, if this oncoproteins is having a mutant MYC gene, this mutant MYC gene will overexpress itself. As I told you in the earlier slides, if there is an overexpression, if there is an excessive amount of a particular gene or particular component, it also leads towards a various diseases. Hence, overexpression is also prohibited. So the mutant gene, that is here, the mutant MYC gene is overexpressing itself. The result of which is the gene amplification. There are more amount of gene of MYC is produced, but these MYC are not the normal MYC gene. They are the mutant gene which proliferate and proliferate. Now, due to which the cancer cells are growing abnormally. That is, they are growing in excessive amount. That we call it as a tumor cells. Now, in all, proto-oncogenes, if they are muted, forms the oncogenes. Oncogenes also produces proteins which are called as oncoproteins. They have the tendency to to mutate the genes, and these mutate genes have overexpression capabilities, and hence their amplification leads towards the cancer cells. So, in all, if you see the normal cells, which are considered to be a good one, a good citizen of the whole body, have transferred themselves and have taken charge of all the proteins. They are taking all the charges, they are taking all the proteins and converting them into the bad proteins or the abnormal proteins. So now we have the normal cell. These normal cells are no longer in normal cells. They have turned themselves towards the cancerous cells. And the growth of these cancerous cells lead towards the genes which are not normal and called as oncogenes. Especially the term genes are also referred towards one of our trousers, what we call it. But here there is no trouser here. The gene is not the normal genes. So hence we need to find the treatment, the remedies for treating those cancerous cells. The happening of these normal cell surface, as we all know, there are antigens which are being present over there. There are tumor associated antigens which are present in the cancer cell itself. So it is very much difficult to identify the cancer cells because they resemble the normal cells. Hence, it is not that much easy to diagnose those cancerous cells. Now, considering the metabolic pathways or the programming or comparison of the normal as well as the abnormal cells, as we did last in the last couple of slides, the metabolic programming of these cancerous cells are very interesting. The metabolic pathways which are followed by these cancerous cells are being reprogrammed. They are reprogrammed from internal factors as well as towards the external factors. The internal factors like genetic mutation as well as the external factors like tumor microenvironment. These genetic mutation, like the change in, as I told you, the change in the type of the gene which are produced, they are not normal. These are the internal factors. That is, for example, p53, as I told you, for example, MYC gene. Considering the external factors like hypoxia is nothing but the oxygen level. The level of the oxygen is not maintained. The concentration of the nutrient is not maintained. The pH of the cell is not maintained. So all these factors, that is especially considered to be the external factors, are not in a normal balance. So they have the ability to produce the abnormal concentrations of all these micronutrients as well as the macronutrients. They lead towards the abnormal metabolism. We have P53, which is not more normal. We have MYC, which is not normal. We have oxygen, which is not normal. We have nutrients, which are not in normal concentrations. Hence, all these abnormal concentrations or abnormal genes, they form an abnormal metabolic activities. Hence, the internal factors as well as external factors combine together to perform the metabolism which is not normal. Hence, they have reprogrammed themselves. They are adapting themselves towards the abnormal conditions. So if you look into the different adaptations which are being formed by the abnormal conditions, that is bioenergetics. Now there is a synthesis of ATP as the metabolism is continuing, is running. There is a synthesis of ATP. There is a production in terms of biomass. The concentration of the nutrients is in full fledged increased in the biosynthesis. Carbohydrates are increased, proteins are increased, nucleic acids are increased. All these concentrations of synthesis of different biocompounds are increased drastically. And the important one is they are also maintaining the redox status. That is, the oxidation is being done, reduction process is being continued. Hence, they have adapted themselves towards the abnormal conditions. Hence, the cells has developed a reprogramming of their metabolic activities. Now, as I told you in the earlier slide, if you see carbohydrate, proteins, nucleic acid, lipids, etc., all these have increased in the biosynthesis. Now, once we have the cancerous cells, once we have the tumor cells, what they are surviving, for which they are surviving, that is some of the compounds which are in the excess amount. Now these compounds can be can be the dangerous, and out of which one of them is the sugar, because the sugar is the component which is required by those cancerous cells. If we do not have amino acid, if we do not have any of the nucleic acids, they will tolerate. But if they are provided the sugar component, they have the ability to divide in more rapid time. It was being conveyed, or it was being discovered by the scientist Otto Warburg, and hence it is called as Warburg effect. Now sugar component is was being found by the scientist Warburg, and it is designated as the Warburg effect. If you compare the two types of cells, normal as well as the cancerous cells, the normal cells take a limited sugar and grows normally. But in case of the cancerous cells, they require only sugar. They doesn't require any of the other. Out of this sugar, they will calculate their own ATP. They will formulate their own different compounds like proteins, lipids, etc. from the sugar itself. Hence, they require more amount of sugar for their growth. Hence, these sugar are important for the growth of the cancerous cells. In a normal way, if you consider the aerobic conditions, that is supply of the oxygen when it is there, there is a release of carbon dioxide, right, in normal course of time, what we call it as a process of glycolysis. But when there is absence of oxygen in anaerobic condition, they go for the process called as fermentation. We all are aware about that, and produces the lactate or lactic acid. So this lactic acid has the tendency to produce ATP, and the carbon dioxide which is produced through the process of glycolysis, it is also producing the ATP. Now, if you realize the amount of production of the ATP in both the in both the processes or in both the pathways, the difference is huge. If you consider the release of oxygen, the release of ATP, which is produced in the case of aerobic respiration, is 36 mole of ATP. That is 36 molecules of ATP per glucose. If you consider the difference, the lactate also produces the ATP. It has the amount of ATP that is 2 molecules of ATP per glucose molecule. Obviously, the cell needs more ATP. If you provide if a normal cell is provided the sugar, it will produce 36 molecules of ATP by following the process of aerobic respiration and performing the process of glycolysis, and produces 36 ATP. Whereas in the anaerobic condition, the lactate is produced and only 2 molecules of ATPs are produced in absence of oxygen. So why there is a difference? Why the amount of sugar is provided to the cancer cell, and these cancer cells go for the process of producing lactate other than the carbon dioxide? Because carbon dioxide has 36 molecules of generation of ATP, whereas the lactate has only 2 ATPs which are produced. This is the sugar which produces the ATP, and the process of healthy cell as well as the cancer cell are being compared in this particular slide. You can easily identify that when the healthy cells go for the breakdown of the glucose molecule, they form the pyruvate, and they are obviously forming the pyruvate and go for the TCA, that is tricarboxylic acid cycle, in the mitochondria, and generate 36 molecule of ATP per glucose molecule, right? And there are no chances or very less chances of following the process of fermentation in absence of oxygen and produce lactate. But in the healthy cells, they will go for the mitochondria and produce ATP of 36 molecule from one glucose molecule. Whereas the cancer cells, these cancer cells go for the production of pyruvate molecule. Generally, in case of what happens in the healthy cells, here also glycolysis is performed, but the pyruvate doesn't produce or does not prefer to go towards the TCA cycle in the mitochondria, but rather than it goes for the fermentation process and produces lactate, which produces only 4 molecules of ATP. Now, if you consider the difference, 36 as well as these molecules of ATP in the produced by the lactate, it has a disadvantage in the cancer cell, that it does not have that much amount of ATP compared to that of the healthy cells. Then also why they are proliferating, how they are proliferating. This lactate has the tendency to go for producing the biomass. The cancer cell does not require the ATP. They are in search of carbon. They are in search of carbon. Hence, this carbon has the tendency to increase the biomass. This biomass increase has the growth of the cell, which proliferating in terms of abnormal cells. Whereas in the normal cell, they require the ATP to perform the different normal metabolic activities. Whereas in the abnormal cells, in the cancer cells, they does not require ATP. If they are requiring ATP, that is the sufficient amount of ATP are generated from the lactate itself. Why they are they require only the carbon. They require only the carbon. Hence, this carbon will help in proliferating for the biomass. So they have two options. Either they have to choose for the ATP synthesis in more number, or they have to choose for the biomass. The normal cell will choose for the ATP synthesis that goes into the mitochondria. The abnormal cells will choose into the biomass production, right? That is, the cancer cell will choose the biomass production. Here in this particular slide, I have shown you the difference. As we know that the process of glycolysis starts from the glucose breakdown and forms the pyruvate. Phosphoenol pyruvate is converted into pyruvate with the help of the enzyme pyruvate kinase. This pyruvate kinase is performing a significant role. This significant role is having of pyruvate kinase is having different exons. Now we are not looking into the matter of introns as well as exon, but you just have to remember that this pyruvate kinase has an a gene, what we call it as a M gene, which is an exon gene. Now, M gene has two different types of genes in itself, M1 and M2. These are nothing but the forms of what we call it as isoforms. It is also referred as pyruvate kinase M isoforms. That is pyruvate kinase M1 isoforms, pyruvate kinase M2 isoforms, and M stands for muscles, or what we call it, which is responsible for the biomass production. So M pyruvate kinase as well as M1 and M2, which is distributed into M1 and M2, they are supposed to grow normally, right? So the glucose, which is dissociated by the process of glycolysis, forms the pyruvate with the help of enzyme pyruvate kinase. This pyruvate kinase has two different types of genes in itself, that is M1 and M2. They require both in normal situation. Now, these normally have an M1 and M2. Now, if you figure it out, in the normal cell, the M1 gene will form an adult cell. Adulteration happens. In M2, the cancer cell is growing normally. In M2, that is, cancer cell is growing normally. Now here I'm showing you the difference where the normal cell as well as the cancer cell, how they are behaving for the pyruvate kinase. If you consider M1 and M2 in a normal cell, the production of the production of M2 is to be stopped at one stage. The production of M2 is to be stopped at one stage in for M2 genes for pyruvate kinase. Here in cancerous cells, M1 as well as M2 are growing normally. They are not inhibited. They are not inhibited. Hence, the cancer cells has more number of adults which are being produced, now how it can be taken, how it can be diagnosed. For understanding the cancerous cells, that is without the M2, we need to know the metabolic of the cells, that is metabolites which are produced by the cancerous cells, they are to be understood. And these metabolites are very much easily shown by analyzed by the mass spectrometry. Now we all have came across the instrument which is the mass spectrometry, which diagnoses or which analyzes different types of metabolites in the cells. So by analyzing it, the pattern of the cells, they are very much interesting. They are rerouting themselves, they are reprogramming themselves to convert the process of glycolysis, which is supposed to form the pyruvate, and this pyruvate has to go to produce the normal ATPs or synthesis of ATP, but other than this they are rerouting themselves. If you see, consider the mutant gene which starts from your left side of the screen, that is the blue spots which are being diagnosed as the mutant gene, right? They are very much less in number, but as the concentration of it is growing and growing, they reroute themselves and number of metabolites are increasing more. These metabolites are having more concentration of carbohydrates, more concentration of lipids, more concentration of different biomolecules in itself, and rather than rather than growing normal metabolites. So they reroute themselves just with the help of the sugar molecules. These sugar molecules go for producing the lactate. They are not depending on the much amount of ATP, but they just require their biomass to generate. And this biomass generation goes for producing different types of metabolites. Hence, the last blocks which you see on your right side of the screen, they are converting them their original metabolites into the specific higher concentration of the metabolites. So hence the overexpression of these PKM2, that is more number of these M2 genes, there's pyruvate kinase muscle gene 2, they result in increased amount of glucose uptake, that is the sugar uptake. They produce more lactate, as well as the autophagy, which is supposed to happen in a normal course, is being inhibited. The cells which are destroyed, the cells which are abnormal, they are to be destroyed by the process of autophagy. They are inhibiting, thereby accelerating the abnormal cells, what we call it as a oncogenes. These oncogenic growth is accelerating by the overexpression of the PKM2. The cancer cells change its metabolism by changing their route. As I told you, that they are rerouting themselves. They rewire their route for growing those abnormal cells. Hence, without the M2 normal cell starts growing, and the cancer cells also grows, rewiring themselves, regenerating those mutant genes, and goes for production of the abnormal cells. Now, as we discussed, that glycolysis as well as the pyruvate kinase are the metabolic activities which happens in the cells, normal as well as in the abnormal cells, that is the cancer cells. But in the abnormal cells, they reroute themselves, they reroute their whole metabolic activities, and forms their own types of proteins for their more division, more number of cells. Normally, the metabolic activities which is related to specific glycolysis cycle, which is shown in your screen, this is only the glycolysis process, but it is not that the cell has to perform only the glycolysis. There are different types of cycles, different types of pathways which are operating simultaneously. So if we have an abnormal effect of this glycolysis, which is producing pyruvate, then what about these many cycles? Because all these pathways, all these generation of different compounds are interconnected with each other. Hence, the process of these metabolic pathways, or the cycles which are prevailing in the cell, will get abnormal functions, will dysfunction themselves. Hence, it is abnormal cells which converts one normal pathway and has subsequent results on other abnormal pathways to be running into the cell. Hence, all these metabolic pathways has resulted into abnormal metabolic pathways in the cancer cells. So the point to remember and to think over is that, as I told you in the earlier slides, there are only 5% chances of cancer cells to be generated from the genes, but there are 90 to 95% chances of the cancer disease to be prevailed by the environment. So we need to think over it, and we need to understand that these cells are to be triggered out of the body by making our immune system powerful. The immune system, as I told you, works on T cell as well as the B cells. We are not going into the detail of the working of the T cells as well as the B cells, but we are just need to understand how these immune system can be grown on a powerful way. There are various strategies of the treating these cancer cells and research strategies. I have just highlighted something over here. That is, identify the metabolic pathways which enable the cancer cells, determine the mechanism that supports the cancer, and to develop the personalized therapy. There are various researches which are going on for treating the abnormal cells. Maybe the chemical therapy, maybe the radiation therapy, but they have its own effect. There is a need to find out the way where the natural processes or natural drugs can be given towards the cancer cells to stop the inhibition or of the apoptosis. Altered cancer metabolism is an exciting avenue for the therapy of the cancer, and there are various types of methods which are nowadays being into the market. That may be experimental methods, that may be surgeries, because cancer cells, if it is growing in a huge form or in a huge mass, that is to be removed first, and then the therapies like chemotherapy, radiation therapies can be going into the followed by the surgery. The different immune system or immune therapy are right now making the immune system very much stronger, so that the T cells can invade those cancerous cells, and the suppressive immune system of the environment of the tumor. We have to stop the tumors to be grown. The immune system of the tumor itself are to be suppressed, they are to be cut off. Hence these tumor cells will be stopped growing if their immune system is being stopped. So two points are to be remembered: making our the normal immune system powerful, and also to stop the immune of the tumor cells. Nowadays, as I told you, there are various types of cancers, various percentages of cancers, and different types of cancer are being diagnosed day to day. The point is, the chemotherapy, the chemotherapy is so dangerous if it is applied on the patient who has the age of about 60 or 60 plus. It can be dangerous, and because cancer kills later, but the chemo kills the first. And but the good news is the therapies are also that much costlier, which kills even more faster than the chemo, right? So we need to understand, we need to rethink our habitat or our day-to-day life, that we are surviving today due to our normal healthy cells. We need to think that make our environment pure, make our habit pure for decreasing the cancer cells. Cancer cells are present in our body. Each and every human being has the cancer cells, but these cancer cells are to be suppressed by making our immune system very powerful. So I thank you for hearing the session, and I hope you have gained some of the information, some of the general information related to the cancer and its metabolism. Thank you for listening. Thank you very much.